Hello everyone. We continue to study the nerve system, and today's topic is the nerve centers and their properties. But first we will consider the kind of inhibition in the central nerve system. As usual, we have the plane and some questions in this plane, a video, and I leave the links to this video and practical work for today in our lab work. On your page in Moodle you can find the new test, nerve centers. And let's start. What is this inhibition in general? In general, definition of inhibition is a state created at synapses, making them less excitable by other sources of stimulation. And synaptic inhibition plays an important role in scalping signals in a nerve system. At the most basic level, synaptic inhibition prevents hyperexcitability. Also, and perhaps even more importantly, synaptic inhibition dynamically regulates activity levels and enables advanced processing and routing of information in a nerve system. And if you don't know what is this synapse, I leave the link to our practical class 7 structure of synapses and mediators, and just you can watch the video in internet. So about classification of the synapses, what we know. According to the signal transmission mechanism, synapses in our and animal organism, we can find chemical, electrical and electrochemical synapses. The electrical synapses usually gives only the excitability signals. Here we see I do this slide less. The mediator chemical transmitter molecule from the chemical synapse can be excitatory molecular, inhibitory molecular, and modulatory molecular. It depends on the structure of the synapses and receptors on the surface of the postsynaptic membrane. At the same time, as you see, the one neuron has many different synapses. Here we see red and green colored synapses, excitatory and inhibitory synapses. And let's start from what happened on the surface of the membrane, excitatory state of neuron. Here I will remind you what happens to a neuron when it's excited. And excitatory neurotransmitters act on it. The postsynaptic membrane of the synapse can respond quickly due to ion permeability, or form a slow response through the metabolic reactions and activations of G proteins. Next one slide, generation of excitatory postsynaptic potential. As a result will be generation of this excitatory postsynaptic potential. And I hope you know this picture. We will discuss it about some practical goal where we study nerve fibers. If the inhibitory mediators act channels allow negatively charged ions to pass through the membrane, or for example it can be the chlorine ions, or sometimes just potassium channels allow the potassium to leave the cells. And hyperpolarization of the membrane postsynaptic neuron gives inhibitory postsynaptic potential. And you know that special molecules in inhibitory synapses work this way, like glycine, serotonin, dopamine, and for example, this I give information about inhibitory transmitters that we see the gamma amino butyric acid, glycine, and dopamine. As usual, gamma amino butyric acid is used at the great majority of the fast inhibitory synapses in virtually very part of the brain. Mainly sedative drugs act by this way through the synapses. Glycine, dopamine has a number important function in the brain. It plays a critical role in the reward system, but dysfunction of the dopamine system also implicated in diseases, disasters of Parkinson disaster. Generation of the inhibitory postsynaptic potential, as you see, but you see this previous slide where the line was up, was moved up. Here we see postsynaptic neuron and generation this inhibitory postsynaptic potential, and this line moved down. And what happened? Brings the membrane potential away from the threshold for generating and action potential, and inhibitory neurotransmitter transmitters hyperpolarize or stabilization, do stabilization of the postsynaptic membrane. This is called an inhibitory postsynaptic potential, and as usual opens the potassium voltage gate channels on the postsynaptic membrane and allow potassium ions to leave the cell. And this way, signal transduction, nerve impulses, way, and action potentials. In general, we have two results from the excitatory synapses and inhibitory synapses. We have, in first case, depolarization, or in a case of inhibitory synapses, we will have the hyperpolarization. Neural integrations, very important function of the nerve system. The net effect of excitatory postsynaptic polarization and inhibitory postsynaptic polarization on the postsynaptic membrane will determine if the net effect is excitatory or inhibitory, and in a neuron it will be if the net effect is more excitatory than inhibitory, and action potential will be generated on the postsynaptic membrane, and impulse transduction will occur or just stop in the different cases. And this slide shows the mechanism of postsynaptic inhibition. What we have the distribution of action potential to the terminal inhibitory neuron, realize inhibitory mediator, special molecule, as you know. The mediator interact with the receptors on the postsynaptic membrane, and two ways. At first, opening the chlorine channels or opening of potassium channels, and just in this case, potassium ions comes out from the cells, or just chlorine ions enters in the cells with negative charge. And in this case, and to these cases, we have hyperpolarization of postsynaptic membrane, increases threshold depolarization, and in the end, inhibition of action. And this slide just describes all this previous one in a text. And what we have just with this, we have, this is a table this picture describing of the work of channels with chlorine and potassium ions and different molecules, and here is the balance of ions and molecules that take part in the formation of potentials in neurons. I think everything was clear up to this point, and let's look at this slide. First, you see two types of inhibition and central nerve system: primary inhibition and secondary inhibition. And the activity of nerve cells can be inhibited without the participation of special inhibitory structures, without synapses. Passimal inhibition in a secondary kind of central inhibition. Passimal inhibition develops in the excitatory synapses, not in inhibitory, without the molecule of inhibitory mediator. In excitatory synapses, as a result of strong depolarization of the postsynaptic membrane under the influence of the nerve impulses arriving to the frequently. Inhibition following excitation. A discrete type of inhibition is that developing in a nerve cells after termination of excitation, and which appears when excitation is followed by strong after hyperpolarization of the cell membrane. And here what we have is this secondary inhibition. Let's look. The activity of a nerve cell can be inhibited without the participation of special inhibitory structures. And the text what we have, and the slide types of secondary inhibition, just passimal or following excitation. So what we have for secondary central inhibition? We just don't have the special inhibitory synapse. We don't have the special molecules. We just have, as usual, excitatory synapse, but the force can be very high, and we have the passimal inhibition or inhibition after excitation. Like in this case, we see this part with low charge, and action potential can move through the inhibitory synapse. Just not so much time. It's time very short. For primary central inhibition, we have two kinds: presynaptic and postsynaptic, and we have some kinds of postsynaptic inhibition, and we will discuss about this now. Here we see the types of primary postsynaptic inhibition, and as we see here, the inhibitory synapses, here it's the black cells, always contact with the body of the neuron. These cells are Renshaw inhibitory neurons. What is this? The Renshaw inhibitory cells. This cells is located in anterior horn or spinal cord, in close association with the motor neurons, and it's an inhibitory cell excited by collateral from the alpha motor neuron to project back and inhibit the same motor neuron, and is the function of nerve centers negative feedback function. And a little bit less we will discuss about this. As you see, these Renshaw cells, we have already met them in the somatic nerve system when we discuss about work of antagonistic muscle. This range cell inhibition, its collaterals from the alpha motor neurons emerge and impinge upon cells called range cells. When the alpha fibers are stimulated, the range cells therefore are also stimulated, and the action of the range cells now inhibited the nerve cells soma of the alpha neurons. This phenomenon is called Renshaw cells inhibition. After Renshaw, who discovered it, the etiology of this phenomenon appears to be to produce a conduction to that even if the corticospinal tract fires repetitively. The frequency of the muscle contraction remains less, and Renshaw cells inhibition lasts for quite a few milliseconds, and thus the muscle is protected against to high frequency stimuli. And Renshaw inhibition from the big-sized anterior horn cells of the spinal cord emerge alpha motor neurons, which and in the skeletal muscles and now upper motor neurons or corticospinal pyramidal tract fibers impinge on these alpha motor neurons. Therefore, then the corticospinal tract fibers, alpha motor neurons are stimulated. And we see this example: just direct inhibition, recurrent inhibition, and lateral inhibition, and one more kind, reciprocal inhibition. Start from the direct inhibition. This kind of inhibition occurs when an inhibitory neuron, these Renshaw cells, realizing inhibitory substance, acts on a postsynaptic neuron, leading to the hyperpolarization due to opening of chlorine ions channels or just potassium channels, and for example glycine at the level of the spinal cord to block pain impulses. And one more, the neurons in the center give off collateral to excite the inhibitory synapses on the neurons in the center. As the neurons become more excited, the interneuron also becomes strongly excited and resulting in greater inhibition of the neurons. This is a negative feedback function which can stop or slow the activity of the neurons in the time and coordinate activities of many neurons in the same nerve center. One more kind, lateral inhibition, is a mechanism that is used through the nerve system to sharpen signal transmission, and this process uses inhibition of the input from the peripheral of the receptive field to better define the boundaries of the excited zones. Motor system and sensory system use this mechanism to focus and sharpen its signals. For example, it is in the eye. And reciprocal inhibition, when the central nerve system sends a message to the agonist muscle, causing movement to contract the tension on the antagonistic muscle opposing movement, and is inhibited by impulses from the motor neurons, and thus must stimulate for relaxing. This neural phenomenon is called reciprocal inhibition, and the theological principles is obvious. When a group of muscles, say the flexor of the elbow, contract the opposing antagonistic muscle, and extensor of the elbow in this example must relax to to this time. And what we have already discussed about a lot of kind of central inhibition. We discussed about secondary inhibition and postsynaptic inhibition. It is a kind of primary central nerve inhibition, and we have only presynaptic inhibition. Looks on this slide. What we see is the types of primary inhibition, and in this case we have presynaptic inhibition. Do you see the difference? See the difference in the inhibitory neuron, black colored cell. It blocks the excitation even before the synapse, before on the fiber. It does not allow the excitatory synapse to be realized, just stop the excitable signal before the synapse. And what we have here we see this slide just better. Inhibitory neuron blocks the signal close to fiber, close to presynaptic axon terminal, but not like in a postsynaptic inhibition. Here we see the body of the neuron, but here we don't contact with the neuron, we just block the signal close to fiber of first neuron, excitatory neuron. And here we see inhibitory neuron, and we have in a synapse we have the same molecules of neurotransmitter, the same serotonin, dopamine, but blocks previously. And what we have, mechanism of presynaptic inhibition. It first occurs at presynaptic terminal before the signal ever reaches the synapse. Inhibition is presynaptic case is the discharge of inhibitory synapse that lie on the outside of the presynaptic terminal nerve fibers before their ending terminate on the postsynaptic neuron, and the inhibitory transmitter realized like an GABA, gamma aminobutyric acid, or other specific effect of opening ion channel, allowing large ions of chlorine or just diffuse in the terminal fiber, and negative charge on this ion cancel excitatory effect of positive charge sodium ions that enter terminal fibril, even just action potential arrival. And in the general we see the same table like in a previous postsynaptic inhibition. We have mechanism of presynaptic inhibition, distribution of action potential to the terminal inhibitory neuron, realize inhibitory mediator, and here we see the blockade of calcium channels of presynaptic membrane of excitatory neuron in a synapse. And we know that calcium ions very important for moving vesicles in a synapse. Excitatory synapse, it just for every synapses it's very important. And depolarization of presynaptic membrane is exciting neuron, and reduce input calcium into the terminal of exciting neuron, reduce secretion of mediator and action potential. Postsynaptic potential, in general we have inhibition of signal, but it's not so easy to understand this all classification. And in general we have the primary inhibition in time, and we use the special neurons, Renshaw neurons. And in general we have protection of case of hyperpolarization and coordination. and in the end, secondary inhibition after excitation. What we have in general, inhibition in central nervous system is an active nerve process which results is weakening or stopping excitation, and inhibition is the result of excitation. You see this, and sense of inhibition, just coordination in central nervous system, coordination of signals and protection of neurons, just all nerve system too. And this mechanism, we have hyperpolarization or depolarization. It depends of the kind of inhibition in a central nerve system, and some structure use the inhibition for give the information or stop information which move in the brain, and very soon we will discuss reticular formation and interneurons of spinal cord. This term some time ago we discussed it. So a little bit more for today. We will discuss about nerve centers in a central nerve system. A nerve center is a group of neurons acting together in the performance of definite reflexes on the or in the regulation of the specific function, one organ or just the organ system. And here we see the neurons of the nerve centers which located on the different levels of the central nerve system. We have the parts on the spinal cord, in a medulla and in a thalamus, and always we have the special center in a cortex. For movement we have the motor cortex place and special neurons here, and one more for regulation of breathing. And even here we see the specific nucleus in the brain, red colored circles, different one. And one nerve centers can have different levels of organization, from the medulla and even the parts in the spinal cord. And what properties of nerve centers we have? Very much the properties of nerve centers. And at first we have the one-way conduction. It depends on the synapse. The movement of the action potential can be only one way, from the presynaptic membrane to the postsynaptic. And sometimes we have a lot of time for movement of the molecules, and light conduction in general in one synapse is very close to the 1 milliseconds. And if we have a lot of synapses in one reflex, this time can be longer. So, summation of excitation. We have two kinds of summation. Spatial, as a result of the integrative function of a neuron, summation of weak signals of synaptic inputs from different neurons to the dendrites and even the cell body of one neuron. And you see here the very weak signals can just summate or not summate, and this neuron just don't generate the action potential and in the trigger zone, and just don't let the stimuli move in fibers. So next kind of summation of excitation, temporary, occur when the presynaptic neuron has some consecutive action potential signals which acts on the postsynaptic neuron, in one neuron and one nerve fiber. In one synapse move very weak signals. Here we see the signals less than need, but if the signals with a different in a short time will move to the body of the neuron, or just to dendrites to the one neuron. In general, these signals will be summed, and in the future it will give the action potential in a trigger zone. So next one, transformation of the rhythm is charge of action potentials frequency after the passage through synapse or neural center, and the biological sense of transformation is amplification of the important signal for an organism, a reduction of an significant signals for an organism, and the coordination activity of two different neurons in a reflector arc. Next one, after action. This is a continuation of the reflex activity after stopping of stimulation. So it's prolonged excitatory potential, postsynaptic potential, and prolonged after potential depolarization, and in the same kind reverberation of excitation. And this excitation move in some circle of the cells where sometimes very long time. Next one, post-tetanic potential and long-term potentiation, amplification of the reflex reaction on a weak stimulus, and the reason accumulation in presynapse calcium ions. And next one, the tone of nerve centers. Nerve centers always in a tonic state. And it is possible thanks to neurons, pacemakers, modulation of humoral factors, afferent signalings, summation, and circulation of excitation. Sometimes we need to know what is this occlusion. The phenomenon occlusion will be that the quantity of excited neurons at stimulation in the stimulate state, simultaneous irritation, afferent inputs of both nervous centers appears less than the arithmetic sum of excited neurons at separated irritation of everyone afferent and input separately. And sometimes we know this phenomenon like facilitation consists in simultaneous stimulation of two groups of afferent fibers at facilitated zone and producing an effect whose magnitude is bigger than the amplified sum, arithmetical sum of those reflexes taken separately. And one more, here we see the stimulation of some nerve cells and the centers, and this phenomenon in the central simplification is characterized by opposite effects of occlusion, facilitation, opposite occlusion. And we have some more kinds, specific function of the nerve cells. As you know, the nerve system is very sensitive to the low concentration of the oxygen, to the pH and level of glucose and even toxins, and the plasticity, possibility to change of function, especially the synaptic function, and Next one, principles of coordination in central nervous system, and we have a lot of principles, and here we see a big slide, and thanks to these abilities to the nerve centers, the nerve system itself coordinates the function of organism, and we already know a lot, but here are a lot new words. Some of them we discussed, and some of them we will discuss. And for example, what is the divergence as a part of coordination function of nerve system? Divergence: one neuron sends impulses to several neurons via its branch axon and can amplify an impulse, and this impulse from the single neuron in a nerve system may be amplified to activate enough motor units needed for muscle contraction, for example, or glandular secretion. So in this case we have the from one neuron to very much a count of neurons. And the new word convergence: neuron receives input from the several neurons. In common, impulses represent information from different types of sensory receptors. For example, allow nervous system to collect, process, and respond to information, and makes it possible for a neuron to assume impulses from different sources and give the answer. For example, the information from 100 million light receptors from the eye converges on 1 million axons and move, you see, convergence, and divergence, which diverge to ultimately influence billions of cortical neurons in the cortex of the brain, and the visual system represented as a neuronal chain. It's very much count of neurons, and in general the principle of common way, it's more for soma, for body, and this principle you see on the picture. One and the same motor neuron involved in many reflex arch from the eye, like was the previous example, from the nose, from the hair, from the skin, from the stomach, from the autonomic nerve system, and effector neuron from the final common pathways for reflexes of the different region can be linked to any of the organism receptors, but like an answer on this stimuli can be just one answer, movement of the body. In the different cases it can be different. The principle of the feedback, we discussed about this. Just look close to this slide. Afferent impulsation generated within the region by the activity of its organ and tissues can be called secondary. In contrast to this first reflex reaction and secondary afferentation, sent impulse in the central nervous system about state of motor apparatus, about state of excitatory gland, and feedback afferentation. In general, we see feedback here, and we see reflector act for first the receptor part, afferent part, central part, and afferent part like afferent nerve fiber, and we move the stimul to the muscle, and we have the back afferentation, back feedback impulses to the horn of gray matter of spinal cord, and as you know, we have special receptors in the muscle. It's in a spindle, muscle spindle. And next one, reciprocity. The phenomena were attributes to stimulation of the nerve centers of flexor muscles channels for example, one leg, inhibition of the center of extensor muscle channel of the same leg, excitation of the center of the extensor key in the other leg, extensor key. Excitation of the center of first group of muscle in a company, reciprocal inhibition of the centers of antagonistic group of the muscle. Do you know this process? And here we see the reciprocal innervation of muscles, extensor, flexor, antagonistic muscles, and collateral branches stimulation gives the inhibitory postsynaptic potential in antagonistic muscle, and the cells of Renshaw helps to give information about inhibitory stimuli, to generate this inhibitory stimuli. Irradiation of excitation, the strong and long stimulation of the nerve centers leads to excitation of the nerve centers, which located close, nearly the first one, and irradiation is stopped by inhibitory neurons. And one more phenomena, success induction. Successful induction. So what we have in this case, the succession of the movements of limbs or other body parts in a pattern of antagonistic reflex action, and walking, for example, successful induction requires alternative flexion and extension of muscle in the lower limbs. And here we see the part of spinal cord must. And one more, the principle of dominant. And we know the scientist Tomsky, and then first part of the 20th century give this dominant is a stable, prevailing source of excitation in a central nerve system, which controlling function of other nerve centers for specific useful result. And the properties of dominant, increasing excitability, the stable excitation attract excitation of other nerve centers to one dominant center, and inhibition of other nerve centers. And the principle of dominant, meaning of dominant, ensures the formation of behavioral reaction, ensures the formation and emotions, participation of the pathogenetics of diseases, and what we have ability to summation, ability to break, inertia, increased excitability, persistent of excitability, persistent. Condition of formation of dominant, we should influence of environmental stimuli, influence of stimuli of the internal environment, for example, the level of hormones or nutrients, and condition of disappearance of dominant, meeting the needs for which form dominant, the emergency of a stronger dominant, and secondary breaking in dominant and can possible. And next one, we have in nerve centers, plasticity and recovery of the nerve centers. What is the plasticity? Is the ability of nerve centers and nerve system to review its connection. And some forms of plasticity are the basis of memory. Other forms enable healthy parts of the nerve system to take over the function of areas that are damaged. Increased plasticity could be useful in many conditions affecting the nerve system, including spinal cord injury and stroke, head injury, and multiple sclerosis. And you have the next one principle, the principle of subordination. And there we have the highest level of subordination, the cortex of the brain. And in the future we'll start to study the brain, and we will discuss about this function of nerve centers and a little bit more principles of force, principles of common way. We discussed about it, and principles of subordination as the underlying departments submits to instruction of an overlying departments of central nerve system. So we have the next part of our practical. It's our lab work about excitement, irradiation of the central nerve system, and a little bit all kind of nerve centers. Nerve centers function. We discussed it all, almost all, and dominant, you see, and what we have, the central inhibition, Sechenow's inhibition, this phenomenon, and lab work for this central inhibition. We have two, as you see, the scientist, the Russian scientist, the Sechenov, and what is the inhibition in central nerve system. What we do, a frog brain is incited at the level of the thalamus, and the cerebral hemisphere is removed. Then the reflex time for withdrawing the hind legs from the solution of acidic solution with different concentration was measured, and the reflex was performed by the spinal centers, and its time indicated the excitability. And then just move the crystal of the salt. Here Sechenov found that application of the crystal of the common salt or a weak electrical stimulus to the section of the thalamus immediately prolonged the reflex time. And from this experiment he concludes that there were nerve centers in the thalamic region of the frog brain producing an inhibitory influence on the spinal reflex. And Sechenov correctly evaluated the great importance of the phenomenon of central inhibition he had discovered, and used it in his theoretical works to explain the physiological mechanism of man's behavior, and where we should to put the salt on the frog brain, and we will watch the video to this lab works, and you see this frog brain. And at first we just do this usual reflex to move the leg in a liquid of acidic, and after putting of the crystal of the salt we just don't have the reflex, because this reflex just stop inhibited. And next one work, just I don't have the video for this work, you can just read the text, experiment of summation in central nerve system. So let's watch the video to the first lab work, irritation in central nervous system and experiment of Sechenov. Just we have the frog without the part of the brain. So we just should to know the time of reflex in a normal state. And after that, remove the salt on the surface of the brain. And then repeat the movement of the leg in a liquid of the acidic solution. And how we see, we don't have the reflex. We see the inhibition in central nervous system, just the reflexes of spinal cord. This reflex absent. For this practical, we have all information on the first slide. You see this video files, and I will give the links to this video on the description to this practical. And here we see the test for this practical work. See you later, next time. Bye bye.